Hello everyone, thanks for being here. I'm Weijie from the Department of Electrical and Computer Engineering at the University of Utah. It's my pleasure to present our research entitled A Miniature Non-Invasive Wireless Tail Cuff-Based Heart Rate Sensor with Motion Artifact Suppression for Real-Time Monitoring of Laboratory Mice.
Here is the outline. I will start with the introduction. The proposed wireless tail cuff-based heart rate sensor design will then be presented. After that, I will discuss the prototype tail cuff sensor's characterization. Then I will present the non-invasive animal experiment results. I will finish this talk with a conclusion. Human genomics is an important medical research aspect which answers big questions such as genetic variation, susceptibility to diseases, gene function, biological pathway, disease's origin, and potential effective treatment. The common approach is using genetically engineered mice as research models. It is critical to obtain long-term real-time vital signals such as heart rate, blood pressure, ECG, temperature, etc. To measure heart rate of a laboratory mouse, currently there are two approaches. The first one is the implantable approach. The advantages are it is wireless and the animals are untethered. Also, it is for long-term measurement. The disadvantage is that it requires invasive implant procedure. The second approach is non-invasive. It does not require any surgery. For example, the tail cuff shown in the lower left corner. However, it requires restraining laboratory animals and is for short-term measurement.
Here is our proposed wireless tail cuff-based heart rate monitoring system. A tail cuff sensor is placed at the base of a mouse tail to monitor its blood pressure pulse waveform. In this zoom-in figure on the right, BP pulsation inside the artery of the tail is coupled to the pressure sensors through the surrounding tissues. The measured waveform is then transmitted through a telemetry circuit to a nearby receiver. A harness covering the base of the tail and the rear portion of the body can protect the sensor and house the telemetry circuit while allowing the animal to move freely. The current research focuses on tail cuff sensors and telemetry system design. Protective harness is planned as next step.
This slide shows the two types of miniature tail cuff design. One sensor design is shown on the left, and two sensor design is shown on the right. The two sensor design is proposed to suppress motion artifacts. The diameter of a typical mouse tail is around 4 mm, so the inner diameter of tail cuffs are chosen to be around 4 mm. The pictures down below show the 3D printed tail cuffs. 3D printing is ideal for quick prototyping. The printed tail cuffs have light weights between 11 to 15 milligrams. Here are the fabrication steps of the tail cuff sensors. The first step is to mount the MEMS sensor onto the PC board, followed by wire bonding. The second step is to coat silicone to provide protection for the sensor and bond wires. Then 4 wires are soldered to the backside of the PC board. The last step is to insert sensor or sensors into tail cuff, followed by additional silicone coating. The weight of the tail cuff sensors is between 14 to 21 milligram. Here are the prototype tail cuff sensors characterization. In the first test on the left, a rubber tube mimicking a mouse tail is inserted through a tail cuff sensor. A reference pressure sensor is placed inside the rubber tube. Then the pressure change inside the tube is manually introduced. The output data from the tail cuff sensor and the reference pressure sensor has 99% correlation. The test on the right-hand side characterizes the step response of the sensor. A tail cuff sensor and the reference pressure sensor are placed inside a pressurized balloon. When the balloon explodes, it generates a step pressure response. It takes about 7 milliseconds for the tail cuff sensor's output to settle and about 5 milliseconds for the reference pressure sensor's output to settle. The equivalent bandwidth of the tail cuff sensor is about 90 Hz. It is sufficient for blood pressure pulse measurement of laboratory mice, which is typically between 5 to 10 Hz. Here's the motion artifact characterization. For the differential motion artifact on the left, a manual motion is introduced normed to the sensor surface. The room square value of the two sensors' output is 1.47 and 2.12 mV respectively. By averaging the two output signals, the motion artifact is suppressed to 0.46 mV room square. For the common mode motion artifact shown on the right, a manual motion is introduced parallel to the sensor surface. The room square value of the two sensors' output is 0.68 mV and 0.42 mV, which is smaller compared to differential motion artifact. Averaging the two output signals results in motion artifact of 0.53 mV room square, which is similar to that of each individual sensor. This slide shows the non-invasive laboratory mouse tail blood pressure pulse waveform measurement setup. We have an anesthetized mouse with tail cuff sensor slipped to the base of the tail, which has wires connected to a data acquisition unit for this test.
This slide shows the measured blood pressure pulse waveform and motion artifact suppression technique. The first row shows the measured blood pressure pulse waveform, exhibiting motion artifacts. For the two sensor tail cuff, the amplitude of motion artifact is around 0.16 mV, which is 3 times smaller than the strong motion artifact detected by one sensor tail cuff. To derive heart rate, second-order derivative is typically used to identify systolic foot, which provides an accurate timestamp of each heartbeat. As shown in the plots on the second row, however, the second-order derivative is sensitive to motion artifact. Therefore, the motion artifact suppression technique is needed. Since the first-order derivative is less sensitive to motion artifact, it is first used to identify the upstrokes, which are shown in the plots on the third row, where the peaks in the red circles indicates the upstrokes. A search-back technique from each identified upstroke is used to determine the correct systolic foot. In the last step, heart rate is calculated using corrected systolic foot position. As a result, two sensor tail cuff design yields higher heart rate accuracy than one sensor tail cuff design due to suppressed motion artifacts. A prototype wireless transmitter module is constructed to demonstrate wireless sensor data telemetry capability. It occupies an area of 18 mm times 14 mm and has a weight about 1 gram, most of which is due to a 0.6 gram battery. 434 MHz phase shift keying transmission with 0.5% duty cycle is used. The maximum transmission distance is about 10 meters. The power consumption is 0.4 mA under 3-volt supply. The transmitter can potentially be incorporated in a protective harness. The prototype wireless transmitter is then connected to a two-sensor tail cuff, which is slipped onto the base of a mouse tail. The measured blood pressure pulse waveform is shown in the first plot down below, exhibiting a heart rate of 722 beats per minute. The second plot is the measured blood pressure pulse waveform using direct wire connection, exhibiting heart rate of 779 beats per minute. The difference in these two heart rate values is due to the different measurement time windows. Here are the conclusions.
a miniature, lightweight, and wireless tail cuff heart rate sensing technology based on blood pressure pulse waveform monitoring with motion artifact suppression techniques is demonstrated. 3D printed tail cuffs and MEMS sensors are employed to achieve small form factor and lightweight. Animal stress is reduced. As a result, reliable physiological data can be obtained. Two-sensor tail cuff design can suppress differential motion artifact to achieve a more accurate heart rate measurement. Prototype technology can enable long-term and untethered heart rate monitoring for laboratory animals. We would like to acknowledge Utah Science Technology and Research Initiative for supporting this research, and Dr. Andrew Zayakivitsky from the Department of Physiology at the University of Utah for assisting laboratory animal experiment. Thank you very much.